Yes, after Dr. Hans' excellent talk, I'm going to move forward on to one of the extreme PCR amplifications, which is known as real-time polymerase chain reaction. This abbreviation, RT, should not confuse you since it's been gotten used to for reverse transcriptase also. So during my talk, whenever I say RT, that means real time. Actually, real-time PCR is a very simple idea. It's an integration of conventional thermocycler with sensitive measuring system. What are the distinguishing features of RT-PCR compared to the conventional PCR? So only these fluorescent reporter molecules, detection and measurements of DNA synthesis basis on these reporter molecules. So what are these reporter molecules? Basically there are two groups. A group of molecules binds the DNA, the double-strand DNA, regardless of sequence. The other reporter group is sequence-specific fluorescent probes. RT system has an excitation light source, as seen over here, that excites the reporter dye. Detectors, seen here, green box, sense the emitted fluorescence, and software does the further analysis. This integration has great advantages on monitoring DNA amplification in real time. And here you see the one of the target specific probes, and I like scorpions, and there's also tagman probes available on that system. When target specific probes type of fluorescent reporter molecules are used, after DNA melting, primers and fluorescent reporter molecules enable the complementary template DNA as seen over here. In that model, reporter molecule complex have two specific molecules as seen here, the first and the second. This emits the fluorescence known as fluorescent molecule, and the other, this one, absorbs the emitted fluorescence and silences it, hence known as quencher. While DNA polymerase works, it is 5' nuclease activity removes and degrades the downstream reporter molecule complex. After the quencher is out of the acquisition, fluorescence becomes detectable to the system sensors. The fluorescence accumulates with each successive cycle of amplification and shown up in a real time as a graph on the screen. So, the other type of reporter molecules are fluorescent dye that specifically binds to double-strand DNA, and the cybergreen is one of the most widely used reporters since simplicity of use and the cost efficiency. It binds the minor groove of DNA double helix, as our dear colleagues mentioned previously, the minor groove of DNA. And fluorescence is much stronger in double-strand DNA than its own single-strand DNA. That's why we prefer this cybergreen instead of ethylium bromide or other DNA-specific dyes. So real-time PCR based on fluorescent absorbance related to double-strand DNA synthesis, and this simple detail creates a great advantage on monitoring and quantification of DNA amplification at each cycle in real time. And that procedure eliminates the post-PCR manipulation obviously, which creates a great another advantage. Of course, there are rules, and as you know here, I'm not waiting you to get all these lines utilized like this. There are rules, we know that. Some same with the rules of conventional PCR, some others same for all RT system, and still some other unique rules for each commercial RT-PCR unit. Basically, they all related to slighted, as you know here, integrity and consistency of the RT-PCR systems, reporter molecules, reaction mixtures, and optimization of all these starting amount of DNA and PCR mixes, and outline target-specific application-related fluorescence signals, which has to be known by the system, and filters has to be fit with these fluorescent dye, and non-specific background clearance is another, the noise removing is another procedure, has to be done by the system or by the users. So there are some rules to be defined to remove of this background, and also to tell the system that the specific amplification starts over that point, which means threshold level. So all these parameters has to be checked before starting the system. We know that it is not easy to become an expert on RT-PCR, but I don't expect you to learn and apply all these requirements. But if we can understand the logic, we can design simple experiments or basic assays, and most importantly, we can able to understand related issues. That's what I want you to do. So basically, two types of analyses are possible by RT-PCR systems: quantitative and qualitative analysis. In quantitative analysis, presence or absence of a target in a template is determined. In quantitative analysis, not only the presence of target, but also the quality of the quantity of the copy number in template is detected. Let's start with a basic prototype of qualitative RT-PCR applications, and you will also practice this in a lab session. This scenario can be named like searching a specific DNA sequence in a given template. Let's say HPV detection in cervical smear. For this purpose, we need to set up PCR mixtures with reporter molecules like, let's say, cybergreen. Remember, reporter fluorescent molecules are only the difference from the conventional PCR mixture. After preparation of a reaction mixture, the second step is how many reactions have to set. A basic rule: for each sample, at least 4 different reactions are required. The first 2 sets are for the DNA template, as seen over here. This duplication is necessary to reduce variabilities due to the copy number and template DNA amount. No DNA template negative control is added to monitor the primary interactions and possible false positive results apart from the template DNA. And last year is used for previously proven positive HPV control. So this experiment is ready to run for RT-PCR as seen over here. After we all set these 6 tubes, now the next step is to set the thermal cycling conditions. This is one of the classic RT-PCR thermal cycler examples seen in the screen. Starts with chemical contamination prevention step which I'm not going to deal with, since it's not our objective today, but it's been used and it's very fantastic to prevent the contamination. And this step followed by the DNA polymerase activation, then cycle repetition starts over here. In this case, denaturation at 95 centigrade for 10 seconds, then unified annealing and expression step at 60 centigrade for 60 seconds, and RT steps ends at that point. The last step seen in this graph is to check the amplitude uniformity, in other words, specificity of the amplitude, which is known as melting curve analysis. As you see over here, the unification of this annealing and extension step, which is one of the unique a procedure for RT-PCR that apart from the conventional, that difference from the conventional PCR system. And after concluding the assay, I mean the terminal cycle is over and the all steps are checked. First an absorbance of the each cycle denoting the double-strand DNA amplification appears as seen over here in the screen. In this graph, dark blue line represents a single level of positive control. The green and black lines are negative controls. Remaining red and green lines are two different deletions of past DNA obtained from cervical smear. Results are ready to analyze. The first step is subtraction of the baseline, which is noise caused by the changing reaction condition and environment. This effectively is seen around 3rd to 15th cycle, when this significant amount of amplification step after this step. The goal of this subtraction is to determine the point that the target amplification is significantly above the background signal, so is essential for accurate measurements of fluorescence. The software automatically sets a baseline, then clears the background noise. After baseline subtraction, the other step, a threshold level should be set to the starting point of significant amplification. Usually, softwares automatically set the threshold at 10 times the standard derivation of baseline fluorescence. However, manual adjustment is also available by users. By subtraction background noise and setting a threshold, the critical addressing of essential data acquisition is completed. Now, software knows what point to start to analyze DNA amplification. The next issue is efficiency. How efficient is this amplification? Especially for quantitative analysis, all reactions should have similar acceptable value of efficiency so that we can compare the results. The efficiency of PCR should be like 90 to 110%, meaning doubling of the amplification at each successful cycle. But in quantitative type analysis, it's not that important, because we are looking just presence or absence of an amplicon. An efficient reaction is acceptable for quantitative analysis. Otherwise, the calculations are not to be that much respective. Now we have an efficient amplification, but is that yield uniform? In other words, is it a specific yield which denotes HPV presence? The other issue has to be solved is this. So during the run, fluorescence changes at each temperature, and it's plotted in this graph. As it is seen here, it is very easy to judge that red and green lines of sample amplification has the same melting profile that the blue positive control line has. The bottom lines are negative controls. Based on this proof, we can make a statement saying the given cervical smear sample contain HPV. After this melting curve pattern or after this melting curve analysis, we can directly make this argument. So, can we design similar experiment by using a melting curve analysis to detect mutations? Of course, but the accuracy of results are still a debate. Fortunately, there are some good news also. So expertise denotes that it is useful to use in screening and multi-care analysis detects wild-type alleles to report negative results in experienced hands. But positive results should be confirmed. Main limitation is same melting temperature, different sequence, as seen over here. So we have all 35 position nucleotides seen over here, G to C, G to A, G to T, and with different energy level, at least two different energy level, and they have minimal variations, but if it is still the same, it has the still same energy level like G to A, G to C, G to A, G to T. So these two lines has quite similar melting curve patterns. So we can discriminate these two mutations. We need further analysis. After the quantitative RT-PCR assay, prototype analysis, now we are starting to another group of RT-PCR assays, which is quantitative RT-PCR. Actually, the most widely used RT-PCR assay is that one, and main objective of this assay is quantification of a target sequence in a template. This quantification also creates comparison advantages. This sort of data also helps to understand the molecular levels of the disease as well as further diagnostic and therapeutic facilities, like gene expression profiling assays, the relative abundance of transcript to determine the gene expression patterns between samples. In genomic profiling assays, the genome analyzes the duplications or deletions. Allelic discrimination assays can detect variations down to a single nucleotide level. Lastly, viral titer determination assays are not often used in anatomic pathology practice and basically quantify viral copy numbers in samples as the name implicates. Northern blot analysis is the gold standard for that quantification, since no amplification is involved in the process. Northern blot works very well, but requires more more RNA, which is usually not available for our daily practice material. From that perspective, PCR methods and in situ hybridization methods are particularly valuable when amounts of RNA are low. However, both of them are only semi-quantitative at best. Various competitive PCR protocols have been designed to overcome this problem, but they tend to be cumbersome. So, as seen in that logic perspective, in Northern procedure, we do not apply the amplification, but in our RT-PCR procedure, the first step is amplification. So this amplification factor determinatively affects the quantization process, so that all steps should be checked and clear that all calculation-based assumptions related to double-strand DNA yield, including reaction mixture components, hardware, and software components, are fully appropriate for this calculation. After that general information on quantification and comparison, we can start to another prototype RT-PCR application, which is QRT-PCR, quantitative RT-PCR. In that prototype, we will see an hypothesis. We will use an hypothesis also. The hypothesis is, let's say, is the EGFR expression level high in a nasal polyp than the non-polypoid, normal-looking mucosa? You do a power analyze and get the number of biologic replica, then this is a statistical step, initial step. Then the steps are listed here, starting with mRNA extraction, avoid genomic DNA contamination, or destroy genomic DNA by DNase enzymes. This is very important for pseudo amplification. After extraction, all controls and test tissue mRNA are ready to analysis. Now, we extract our DNA and it's in our hands, and the yield, the amount is enough to test, and we start. We want to understand that whether or not EGFR expression is high in nasal polyp stromal cells than normal polyp mucosal cells. Non-polypoid mucosal cells. We are going to compare the amount of EGFR mRNA expression in two groups. We need an internal control gene expression level as a standard. In this experiment, RPLPO housekeeping gene, we select this one. We use different deletion mainly because of efficiency, polymerase chain reaction efficiency, and comparison measurements of outcome DNA yield. We use a reference gene to avoid the relative expression differences. Choosing such a gene for normalization is another important issue, and in summary, this gene has to be expressed in all cells in approximately the same level, and in parentheses, medium level expression is advantageous, no less and no too much expression is advantageous to compare the other system or the other expressions. Now, the one is we put all these sat in our RT-PCR machine, and thermocyclers started to work, and the run is completed. We have an amplification graph over here as seen in that screen. Standard performance and quality parameters has to be checked at set as first like there is no contamination, no amplification seen in no template control, baseline saturation and threshold level are automatically set, and melting curve are uniform. And reaction efficiency is acceptable, like 0.99. And the results here is the persons of each step at each cycle. Let's start the group and analyze the data. So this is very complicated, but we are going to subtract the other lines, and we only meet with our initial lines, which we need to understand. For example, for reference gen, RPLP, polyps reveal some level of expressions, and expression levels are quite prevalent to deletion ratio. There are only one cycle difference between each deletions as seen over here. You see the three different reference level, and each of them has one cycle difference in between. That means they are quite parallel, and the efficiency seems quite good. And the expression level of RPLPO gene in normal-looking mucosa reveal one single difference in between each deletion series as similar to polyp pattern. Now we merge them, normal and polyp. The expression level of RPLPO is normal-looking mucosa reveal one cycle difference in between each deletion series as similar to polyp. Comparison of the reference gene, RPLPO expression levels to polyps and normal mucosa is seen here. It is easy to see two difference in between same deletion levels, which is one cycle and denotes two folds. So as a result, statement indicated in this graph is the reference gene RPLPO expression levels is twofold high in polyps than the normal-looking or non-polypoid mucosa. And for the target gene, EGFR, as seen in this graph, polyp level reveals more copy numbers than mucosal samples. Please note that blue line stars before red lines. and the graph between same dilution levels are quite equal, which is 10 cycle, and 10 cycle means 10 folds. So, as a result, statement indicated in this graph, the target gene EGFR expression level is 10 folds high in polyps than normal-looking, non-polypoid mucosa. Here is software calculation results. EGFR expression in polyp stromal cells is 5-fold of non-polypoid mucosal cells. This was one of the very simple example of qPCR application. As you see the acquisition, the fluorescent absorbance of target gene and reference genes are 5-fold different, so you can say the difference is 5-fold. And this goes like more recently, it is possible to use more than one color probe in each well. As far as we know, 5 different color probes in a single vial or well is detectable with today's technology. In addition, well plate technology also enlarged the plate capacity, reached over like 3,000, more than 3,000 per plate. From that point, technology enabled the scientists to handle 3,000 times 5 equal to 15,000 gene expression level detection in a single plate in less than 2 hours. I would say this is a great opportunity to understand pathogenesis of disease and will help diagnosis and treatment opportunities more recently, more efficiently. And this is how RT-PCR works. Thank you for your attention. Now